I'd also like to thank our faculties for their continued support in helping us and facilitating this event. Dr. William Xiao, Dr. Amy Lee, and Dr. Faraz Hosh. And here are the upcoming VanBug event dates. More details will be announced later. So if you don't want to miss out on any events that we host, you can also subscribe to our mailing list by going to our website and going to the contact tab and filling out the mailing list form. We also have a public calendar as well as social media where we promote other events in our network. And there's also our YouTube account where we upload the seminars. Lastly, if you would like to suggest a speaker for future seasons, you can also go to the same page and click the suggest a speaker link, and it will prompt you for to fill out a speaker you're interested in hearing from, and we will try our best to recruit them. VanBug is sponsored by the Graduate Studies in Bioinformatics at UBC and SFU, Genome British Columbia, the SFU Big Data Hub, UBC Michael Smith Laboratories, Langara, Stem Cell, and the Vancouver Prostate Centre. We're also affiliated with the ISCV, as well as our sister groups, MomBug and Torbug. For the Zoom audience, please ask your questions in chat, and a VanBug volunteer will voice it for you. We will first begin with our introductory student talk. Julie Sukar, master student in bioinformatics at Joy Lab. And their talk is titled Closing the Transgender Gap in Health and Research. Afterwards, we would have our feature talk from our keynote speaker, Ewan Gibb. He's an assistant professor at the Department of Urologic Sciences at UBC, as well as a senior research scientist at the Vancouver Prostate Centre. And his talk is titled From Fungi to Bladder Cancer, Decoding the Non-Coding for Biomarker Discovery.
Hi, everyone. I'm Julie Sukar. I use they, them pronouns. Let's see if we can figure out this clicker. There we go. The talk name changed. My bad. So we'll be talking about trans inclusion in bioinformatics research. So just a quick backgrounder of what transgender means. There's a lot of misinformation and disinformation out there about it. So when we think gender, we usually think of these two most common modes, cisgender woman and cisgender man. Cisgender just means the gender is what you'd expect based on the body. Now, if someone who we thought was a woman actually understands themselves as a man, they feel they would be much happier that way. He can take the choices to transition his body, his name, his pronouns, and become a man. That's what we call a trans man. Similarly, someone can understand themselves as a woman, and she can make choices to change her body, name, pronouns, and that's what we call a trans woman. Trans just means a certain kind of person who transitioned. And anyone can understand that they're better off as both or neither or something that flows or something in between, like myself. That's what we call non-binary, just meaning not in the binary, as in man, woman, it's a binary state. Anything outside of this binary state would be a non-binary person. So I made the 5 modes framework to make this a little simpler in terms of designing systems and designing research. So instead of thinking of the 2 basic types of gender, which would be man, woman, we have to think of at least 5 kinds of bodies. So that would be cis men, cis woman, trans woman, and non-binary person. These symbols here to help understand, they're not like official symbols for trans people. It's just a visual aid. So let's give them names to humanize them a little. Max for cis man, Oliver, Dana, Emma, and Sage. And the point of humanizing them is that now our brains has an easier time applying characteristics to these people. Now, Human diversity is quite huge. So these are just the most typical characteristics. These are physical characteristics people usually aspire to or move towards in their transition process. So when it comes to men, we have a cis man and a trans man. There's lots of characteristics that are very similar, some that are different. Same with everyone else. And the big thing here is when you design databases or clinical staff, you can know how to include who. So if you're doing a chromosomal sex chromosome study, you might want to include trans men and non-binary people who have XX chromosomes. Well, but how do you know if a non-binary person has XX chromosomes? You ask. So for any of these things, whenever there's variety, you need to ask those things specifically. Whereas before, when we were only considering cis people, you can just assume that gender and sex are associated. You can assume certain kinds of chromosome setups or body configurations. We don't get to do that anymore. Whoops. Okay, well, I just do computer stuff. Why should I care? Well, the computer stuff you do matters a lot. So first is a lot of computer stuff affects patient interaction. When you create forms to recruit people, you need to collect gender respectfully and completely. If you don't collect gender appropriately, you're going to miss out on a lot of people being represented properly. You need to collect preferred name and pronouns, ideally, and transition history if it's relevant. Say if you're dealing with prostate tumor, and you have a trans woman with a prostate tumor, you need to know when she began transition, what kinds of transition she did, so you understand better the life history of that tumor and what kinds of sex hormone impacts that tumor has been exposed to. When you make recruitment material, guidelines, educational material, try to make it gender neutral. Where that is not possible, create gender neutral alternatives. It's okay if you still use gender stuff to recruit for things related to gender and sex, but have some alternative so that trans people do still get to engage with your material, be recruited into your studies, be understood by your analyses. Whenever also sometimes a lot of bioinformaticians make health systems, hospital data systems, databases. And when that happens, there's a lot of issues for trans people when it comes to outing and misgendering. So outing is the issue where you are trans, you didn't tell anyone, and then someone else tells other people. So if you have hospital wristbands, this was a scandal recently at VGH where, I think it was VGH, where you have the hospital wristbands and it would say like, oh, trans woman, this is the gender that we have for you in the database. And people are kind of horrified because, well, if you're outing people as trans every time, there's plenty of clinical staff or custodial staff or whatnot that you don't want them to know that you're trans. Because sometimes people knowing you're trans invites a lot of aggression. So outing is a thing to avoid. Unfortunately, because of transphobia, trans identification is a sensitive data in a way that gender isn't normally considered sensitive because it's usually something you expect to be able to just see. Misgendering is the opposite problem. So I myself am non-binary, I use they, them pronouns. If I'm interacting with a doctor, when I go to a hospital, every person I talk to, I have to come out and explain my pronouns. Otherwise, I'm going to feel very uncomfortable in that interaction. And so when it comes to misgendering, it's the opposite problem where you want more disclosure and disclosure made easy so that people know how to engage with you. This is the case of Ray Spoon. Ray Spoon is a famous musician here from BC, well, local famous. They are non-binary and they had cervical cancer. They made some amazing articles about it in Medium in 2020, talking through their journey. So because cervical cancer is very gendered, there's all of the systems around it assume that the people who have it are women. Ray Spoon was misgendered constantly during the experience, had a really hard time getting doctors and staff to take the misgendering seriously as a problem. And as a result of those issues, had a lot of thoughts of suicidality and of avoiding care. It's a well-documented pattern that if people are misgendered in clinical interactions, they avoid care. And this is even for not taking HIV medication, not taking care of themselves. So this has severe consequences for trans patients. Also, when it comes to data analysis, you're not even engaging with patients. You are going to be engaging with databases. And if you've ever seen patient databases, they collect gender and sex at birth separately. And that's because some people's gender change. The sex at birth is fixed. So you collect sex at birth because that teaches you a little bit about the person's body and physiology, but gender is actually more important. If you only collect sex at birth, you have no idea what this person's gender is now. And you would be misled about this patient and who they are. Gender is not sensitive, trans identification is. So sex at birth data is now sensitive because if someone has both, they can understand if you're trans, because then your sex at birth doesn't match what you'd expect. When you're labeling tissue samples, those kinds of things, things that have already been removed from the patient, if they are trans and have transitioned, the hormone replacement therapy does affect those tissues. So like, how would you label the breast cancer of a trans woman who's had hormone therapy for 20 years? Would you label it as male or female? Any takers? You could try to say male because that's the XY chromosomes, but XY chromosome matters way less for cancer development. The microenvironment, the tissue, the fact that it's a breast that developed in that way, the fact that it was exposed to estrogen for 20 years, way more relevant. So you can't quite just say male, female. You'd have to explain, well, male at birth, transitioned, this is how many years on feminizing therapy this person was on. Sex for trans people starts to become more complex because of the transition process. And so it takes us understanding this a little more deeply to be able to record it properly in research. Often with trans patients, we have small sample sizes, so I would recommend grouping by function. If you're studying menstruation, it doesn't matter if some of the people in the cohort are men, you can still group them with the other people. And then if you see the samples, well, if you report specifically the trans samples, if you do a sub-analysis, if it doesn't differ significantly from the rest, you can say, well, looks like it groups well enough. And because we are so understudied, reporting all the data you have on us is very helpful. Also, studying trans people can have some advantages for your science because we're understudied, it's high impact. And because we change hormones and things like that, you can isolate embryologic and hormonal effects in an ethical way, which usually you cannot. So let's do, with the last minute I got, a little bit of a case study. So this is one of the unfortunate ways that people have been recommended to collect sex and gender in an ethical way. They do a lot of things right, but there's like one specific bad thing that they're doing here, and it's not super obvious. But try going through this as Oliver, the trans man, or as Emma, the trans woman, and let me know if you see something. Yes? And why is that a problem that it says transgender on question 2? Exactly. That's not what you call yourself. It's one element of your identity, but it's not your gender. Perfection. Any more comments on this? Okay. So, well, first off, saying prefer not to answer is great. It gives people agency. Love that. They missed intersex. That's also a sex category. Intersex is not trans. It's just your body develops with bits of both. And so it is a different sex category. Gender is woman and man, not male, female. And when you say transgender, if you are a trans man, you don't know which one to pick. And so people pick whatever they feel right, but they also don't trust you anymore. They're no longer trusting you to engage with their data ethically or in a well-informed way. If they changed it to non-binary, that would fix it. Thank you so much for listening, and I hope this helps you include diversity into your research better. Oops. All right, we have a little time for questions. Any questions? Yep. Yes, AIN is their acronym for Indigenous. Yes? Very good question. So gender is somewhat cultural also. Gender is a kind of person, it's a category. And different cultures will have different gender categories. So two-spirit is the term that North American Indigenous people use as the umbrella of the different unique gender identities that certain Indigenous cultures have. So someone who's two-spirit is someone who you might call queer or non-binary in Western culture, but there's specific social roles, characteristics, behaviors that are unique to each culture. And so they each have their own names, and then two-spirit is the overarching term for all of those. So because there's this cultural racial element, it's a separate kind of identity. Thank you so much.
Sure, you're set up pretty quick, but do you want me to read out your bio?
Sure, yeah, that's nice.
And do you want them to ask at the end, or if you see them asking in the middle, would you?
Whichever works better, honestly. I think a little engagement's good. Yeah.
Okay. Yeah. Okay. So to introduce Dr. Ewan Gibb again, he is an assistant professor in the Department of Urologic Sciences at the Loud. Oh, okay. It's connected to the computer though. This isn't. Is this working? Dr. Gibb is an assistant professor in the Department of Urologic Sciences at the University of British Columbia and a senior research scientist at the Vancouver Prostate Centre. His research program focuses on the discovery and clinical translation of molecular biomarkers in bladder and prostate cancer, with an emphasis on long non-coding RNA, transcriptomic, classifiers, and digital pathology. Prior to joining UBC, Dr. Gibb worked in industry developing genomic tests for cancer care, experience that informs his translational approach. His laboratory integrates molecular biology, computational analysis, and clinical research to improve risk stratification and guide treatment decisions in genitourinary oncology. Thank you.
Thank you for the lovely introduction, and thank you for hosting me today. Yes, sir. You have nothing on Zoom? Oh, no. Let's correct that. Can't make this thing go away. Is that okay? There we go. That works. Not on Zoom. They shared it. Should I just share my desktop, maybe? Yeah. We're good. Okay. I was gonna say thanks for everyone for coming to this and all that bits and pieces. Yeah, so you can read my title, my talk. I'll just jump right into it. So really the lab vision that I have presently is to develop biomarkers for both bladder and prostate cancer. And the intent here is both diagnostics, so like prognosis, and also predicting treatment outcomes. And what we really want to do is not just develop a biomarker, but we want to actually see this implemented into a clinical setting. I'll tell you a little bit about how we see this working and how it actually works in actual real practice. I have like about 8 or 9 years of industry experience doing exactly that. I'm hoping to build a lot of this program out at the UBC as well. So I want to take you for a little journey, going way back to my undergrad days. And this is where I started working in biomarkers. And the point of the story really is to show you that where I came from, my sense of where I started in urologic research, but also to show you how non-coding RNAs can actually be biomarkers in diverse systems, and to show you how effective they can be using very simple methodologies. So in the beginning, I worked on a disease called Dutch elm disease. So this is a tree disease that's caused by a fungal pathogen. You know, you don't have to be a botanist to see that the sick trees on the left there and the healthy trees on the right. It causes a lot of economic damage or ecological damage in Canada, so it's quite significant. We use Dutch elms a lot for shade trees. What it causes is this little fungus called Ophiostoma ulmi, and you can see here this little black fruiting body here. That's actually how it disseminates spores. And as if that wasn't enough, you know, the tree gets these little Dutch elm bark beetles that come along and they burrow into the tree bark and they disseminate the spores around. So the tree gets really hit twice. It gets the beetles and gets the fungus. And so, of course, you're probably sitting there, this guy does cancer. What is he talking about fungus for? Well, there's a lot of parallels in fungal biology and cancer biology. And so different strains of the fungi actually have different virulence rates. And the problem there is when we do strain typing, is, you know, these things look largely the same. So they might grow slightly differently. They might look slightly different on the microscope, you know, but in the end of the day, it's very difficult to distinguish them. And a lot of cancer is very much the same. You'll take a prostate tumor, you'll look at it, and someone's like, oh, that looks like bad prostate cancer. And someone else says it looks like a bad prostate cancer, but in truth, it may not actually be bad at all, or vice versa. And so this begs the question, can we then identify some biomarkers to help distinguish these various fungal strains? And this is very, very rudimentary, basic even. I think a lot of you might recognize this type of gel. This is a PCR gel that I did in my undergrad days. But what you can see here is that the different strains of fungus actually can be differentiated by a simple PCR assay showing two different band sizes. And so the parent strain, the Ophiostoma ulmi, has a larger band, which indicates like a bigger PCR product, whereas the more aggressive strains have a smaller one. So now we have a sort of an indicator of like what could be aggressive and what could not be aggressive. And what we found out that was made this particularly interesting was that the difference is actually caused by this self-splicing intron, a group 1 intron. So these are very unique. They can actually splice without the benefit of proteins. So they can actually self-catalyze their excision from an RNA strand with presence of magnesium alone. So that's the non-coding part. But the plus minus size actually was due to what's called a homing endonuclease. And so these are little mobile DNA parasites that actually catalyze their own dissemination across genomes by having a very sequence tolerant, but it's very specific catalytic site for fraction mobilization. So they basically will cut and then recombine into new genomes. So that was kind of the start of where I got into biomarkers. Now in urologic cancers, you know, we're looking at a totally different spectrum of disease. We're asking a very different question. Now it's not about virulence in pathogens, but now we're looking for prognosis and treatment guidance. So how can we help patients, right? So that's kind of what we're trying to do. And so my research program largely focuses on both bladder and prostate cancers, but you know, thinking about delving into kidney when the time arises. So since my life is basically bladder cancer for the past decade, I'll talk to you a lot about that over prostate, but this is why it's a bad disease. Okay, so bladder cancer, it arises in the transepithelial layer lining the bladder lumen. It's a fancy way of saying the inner skin of the bladder. In the US and Canada, it's about the 4th most common in males and 12th in females. So it's quite a lot, quite a high prevalence cancer. It's three times more common in the men than in females. And the median age of diagnosis is 73. So the majority of people in this room are totally fine, not to worry. You know, some of us maybe not so much. Unfortunately, it's a very aggressive disease associated with high risk of morbidity and mortality. So this means it causes a lot of pain and it can kill you. It's also very expensive. And part of the reason for that cost is that it's requires a lot of high monitoring. And so that creates a lot of issues for the patient because they have to keep on coming back to the clinic. The doctor has to charge them for these for their treatment. And this is kind of an economic problem in addition to a health problem. So a little bit of the basic bladder biology. So you probably recognize this. This is a crude cartoon of a bladder showing the ureters and the kidneys. So the bladder basically is a fluid-filled sac surrounded by muscle tissue and a fat layer. The actual bladder lining itself is very thin. It's only about 3 to 6 cell layers. And I'm not going to go into the details of those cell layers for this particular discussion. But it's important when we're considering non-muscle invasive versus muscle invasive bladder cancer, because these are actually two different diseases. Even though you'll look at them as bladder cancer, they're actually treated clinically very differently, and they have different rates of mortality. So in the US, we're estimating approximately 80,000 patients will come around this year alone. The ratio is kind of interesting. Non-muscle is a little bit more prevalent. About 75% of patients will present with non-muscle invasive bladder cancer, whereas 25% with muscle invasive. This matters a great deal because non-muscle is a little bit more benign. I'm saying benign very loosely, versus muscle invasive, which is nearly always fatal. So getting a little bit more granularity, and this is going to come important later. So if you remember anything, try to remember bits and pieces of this slide. So on the non-muscle invasive side of things, we have what's called TIS or CIS. This is carcinoma in situ. These represent flat lesions looking a little bit more like a red inflamed part in the bladder lining. Moving up the spectrum of TA. So TA is a little less aggressive than the carcinoma in situ, but they look like little jellyfish. They're like little papillary structures. They haven't invaded into the bladder lining at all, as you can see from this diagram. And then you have T1 disease. This is kind of a gray space. So T1s can be very aggressive or they can be relatively non-aggressive. They can be easily resected, but they're quite difficult to treat. And there's a lot of research and clinical trials ongoing for new therapies for T1 disease. And then things get worse as you go into muscle invasive bladder cancer. So these ones are the nasty ones. This is when the tumor tissue is actually penetrated through the lamina propria. So it's actually penetrating into the muscle layer. And the deeper you go, the worse it gets. So if you have a T3 or T4 tumor, for example, you're gonna have a lot more problems than if you have a T2. T2 and T1 and below can all be treated simply with surgery, meaning they can remove the bladder and cure a patient. Not universally true, but in generally. However, muscle invasive requires much more aggressive therapy, usually systemic chemotherapy. So that brings me to management. So management, and again, this sucks. This is a really crappy disease to get. So management for non-muscle invasive bladder cancer is primarily based around recurrence and progression. So recurrence, this is where we create this healthcare issue, right? So patients will come back again and again because we always have to check to see if their bladder, there's been a recurrence in their bladder. At this point, we're leaving their bladder intact. We don't want to take that just yet. There is a somewhat of a risk of progression, and this means that the tumor will actually advance to a higher stage disease. So if it was non-muscle, it can actually become a muscle invasive tumor, and then a whole different set of clinical parameters come into play. Different therapies come into play as well. So unfortunately, diagnosis involves this rather unpleasant procedure where they put what's called cystoscope into your urethra, and they actually examine with a camera the inside of your bladder lining. And if they see a lesion, like a tumor, they will resect it with a procedure called a TURBT. And this just literally means they'll take a chunk of the bladder wall alongside the tumor. And this works for two purposes. First is to diagnose, and second is they can actually use this as a treatment. You debulk or you remove the tumor from the bladder of the patient. After this, they'll receive what's called intravesical therapy. So they'll actually do an installation of chemotherapy into the bladder. So they inject this drug into the patient's bladder, and you have to hold it for a while and then release it, void it later. If it's a particular higher risk, say T1 disease, they'll receive what's called BCG. So this is a attenuated bacterium that can actually activate the immune system and causes the immune system to attack the bladder tumor as well. Response rates to most of these drugs is pretty medium, say about 40% of them will respond. So they require additional therapy and or if it's really bad, more radical therapy. So muscle invasive is much, much worse. So now we're not thinking about recurrent, or we're not thinking about recurrence and progression in the same sense. Now we're thinking about recurrence as in local distance metastasis. So this means tumors come back either in the void space where the bladder would have been, or it can be a nodal involvement, or it can actually be a distant metastasis. And progression likewise is progression means advance to metastatic disease, which is problematic. So again, it's not very nice, but to treat muscle invasive bladder cancer, most patients will ultimately lose their bladder. Bladder sparing therapies are being explored, but there's not a lot of data supporting this is actually very beneficial to the patient. We can't cure if we leave the tumor in place. There's a great risk of recurrence. So what they'll typically do is they'll do what's called neoadjuvant chemotherapy. So they'll do about 3 to 4 cycles of cisplatin-based chemotherapy prior to having their bladder removed. As you can imagine, platinum-based chemotherapy is also very aggressive, and these are elderly patients, so their tolerance for this kind of treatment is not that great. And then after that regimen, of course, they go get their bladder removed. So you can imagine this is very, very unpleasant disease for people. In select cases, they can do what they call bladder sparing, bladder preservation, bladder sparing. This typically involves a maximal T or RBT, so they'll cut as much tumor out of the bladder as possible, and they do a combination of chemotherapy and radiation simultaneously. This is more for select patients who can't tolerate their bladder actually being removed via surgery. And there's a lot of advances working in the field right now of moving immunotherapy and other agents such as antibody drug conjugates for more advanced or relapsed disease typically, but we are trying to move it to the adjuvant setting. Okay, so that's the background. That kind of leaves us with what bladder cancer is and why we don't like it. So when we're thinking about biomarkers, and this is kind of where my world intersects with the machine learning world into clinics, is how do we go about doing this? And so the pathway from discovery to clinic is actually very long and very challenging, and there's a lot of places for things to go wrong, and there's a lot of challenges along the way. But more often it starts with discovery. So we're trying to figure something out. We have a clinical question we're trying to answer or try to help a clinician answer. And so the phases there is after discovery, say we have a nice biomarker, we think it's pretty good. We're going to start doing something called analytical validity. And that just makes sure that our test is repeatable in the actual lab. So when we're giving someone a result, that the results are consistent across users and across platforms. And of course, clinical validation is of utmost importance. We need to make sure that our test reproducibly produces the same result no matter what the conditions we're throwing at it. And then once we get through all that, then you have all these regulatory hurdles, which is like the FDA, the CLIA, the New York State certifications that you have to pass through in order to actually have your test be marketable or actually just released even in a clinical setting. And then you have to integrate your test into the clinic. So you have a physician who's, say they want their answer in 2 weeks, but your test is done in 4 weeks. Well, you're not really helping anybody, right? So you have a turnaround time you have to take into consider sample type. Does the clinician even routinely collect your analyte? If it's blood and they don't collect blood, then you're kind of dead in the water. Hopefully you thought about this at the design phase. And then, of course, in the end of the day, we have the challenges and outcomes with reproducibility and demonstrating real-world benefit and adoption. So this is just once you even have a test on market or even in a clinical space, often clinicians don't even want to use it because they're not convinced that your data is strong enough, or they're just biased towards their own intuition of how they treat a patient. They just don't use this information. So there's a lot of hurdles. So when we sit there and we say, okay, what are the clinical challenges? When I'm thinking about biomarkers and when I first started UBC, I thought, okay, what's my first problem I'm going to answer? I try to answer. And this Brent Inman here, I think he's a colleague of mine. He's a urologist at Western Ontario right now. He says in this tweet, a test to be valuable has got to change what I do. And this is the clinical mindset, right? These are what people think. When a clinician comes to work, they're like, okay, I'm going to do things according to standard of care, according to protocol, whatever guidelines they're using. If you want to change their mind, you have to give them something that actually is going to do something different for them, make them make a different decision. And so really the key considerations that we think about with biomarker development is the clinical relevance. So like, am I providing an actionable insight? Is if I give them a result, are they going to say, okay, I'm going to do something different. I'm going to treat more aggressively. Or I'm going to withhold the treatment. I'm going to just change my practice, right? Then we're going to do something called model evaluation. We're going to look at the performance by what metrics though? Are we doing AUCs? Are we going to do like a biological validation, a clinical validation? Like how are we assessing our performance of our biomarker? Using the cohorts or data sets we have available. And then we're going to start thinking about features, like what am I even going to choose? Like, what set of genes do I use? Do I do like imaging? Am I doing histology imaging? Am I doing proteins, or am I doing RNA? Like, what do your feature selections look like? And finally, interpretability. Like, we can make the most complex, beautiful machine learning algorithms, and they spit out this code that nobody understands except for ChatGPT. And then, the clinician gets it and they're like, well, I don't know what to do with this. And believe me, clinicians are extremely intelligent people, but they're also very dumb people because they want it black and white, because they live in this high intensity environment. They need decisions to be easy. Tell me what to do. It's a very weird thing to say, but they'll all agree. It's very strange. So I'll tell you a story about proof of concept. So to give you a sense of like time here. proof of concept, show you why I think non-coding RNAs are valuable biomarkers and sort of give you a sense of where this all came from. And so this takes me back to my days at GSC. I was working with Anash Birol, and I got involved in several TCGA projects, including Bladder, working with Gordon Robertson, one of probably the best analysts I've ever had the pleasure of working with in my life. And Seth Lerner was probably one of the lead minds in bladder cancer research in clinics in the US. And we published this paper on muscle invasive bladder cancer. And the long story short here is we did some clustering and we identified 5 novel subtypes in the TCGA data set. This was heavily based on mRNA gene expression, which at the time was pretty conventional. Each of these subtypes had sort of unique biology and clinical characteristics defined by their gene expression profiles. And really the point of this is just to show you there's 5 groups and there's some structure in the data, right? So we don't really need to take much more than that home. But they also had unique prognosis, and I don't know if you guys are familiar with looking at KM plots, but basically, I don't know if you can see this on the screen, but the sort of the way the curve flows is like if it's higher up, that means the patients are doing better. If it drops down towards the bottom, that means they're doing worse. So what we're looking for is large separations between these curves. And I know you guys all know what box plots are, or so then aside on B, you can just see basically that they had different levels of purity. So there's different characters to these subgroups that we've built with TCGA. But unfortunately at the time, TCGA was like, it probably had about maybe 10, 15 TCGA projects ongoing. And the struggle with TCGA at that point was novelty. They're starting to say, okay, what's new? This is getting boring. You're just doing these repeat cancer profiling stories. And so I happened to be working on non-coding RNAs at the time, and Gordon approached me. He said, hey, you're doing some really cool work non-coding RNAs. Do you want to work with TCGA Bladder? And I said, that sounds pretty rad. Let's do it. And so we started doing some work with non-coding RNAs. And so to give you a bit of background, what non-coding RNAs are. So these are mRNA-like transcripts, about 200 to 2 kilobases in length. They can be a lot more. They don't have open reading frames of any significance, and they can be polyadenylated. So they look a lot like mRNAs. They're just not translated into proteins. And the reason I put this little tube here is because the cut point for 200 nucleotides, defining them as long non-coding RNA, is basically the exclusion limit for the silica columns that we use when we do RNA extraction. So it's kind of very historical, but I thought kind of cool. So the human genome is actually, as you're probably very well aware, like largely composed of non-coding RNA or just junk DNA, as they say. But what's very interesting is these link RNA expression patterns tend to be very tissue and cell type specific. They can exhibit differential expression across a number of different tissues and under different conditions, so they can actually shift in response to treatment or to stress. But this variability really suggests a unique potential as biomarkers. And I think you guys are well aware where I'm going with this. But they can also serve as potential therapeutic targets because they may have some functions. Functions, I really don't want to belabor what their function is because I'm not entirely convinced they are all functional. I think a lot of it is actually transcriptional noise, but we can definitely leverage this for biomarkers. Okay. So what I did with TCGA there is we did sort of a clustering based approach here. And again, we came up with a 4 cluster solutions. We have these unique groups. And really all I really want to take from this is that we took one of the tumor subtypes that TCGA found with their subtyping, and we split it into 2 different groups. And what was interesting about the 2 different groups is one was depleted in TP53. So this is one of your tumor suppressors and enriched with FGFR3. Mutations. So that was actually quite striking, and also that they were very prognostic. So we're using non-coding RNAs to repurpose TCGA, and we found something quite different. And then at that point, I'd left the GSC and I moved on to a company called GenomeDX, which was then Decipher, which is now Veracyte. So it kind of went through a lot of changes. But one of the first things I did, when I got there, was I wanted to start developing a classifier based on that work, because I said, okay, we've got this interesting subgroup of patients that do incredibly well with just standard of care therapy. Let's see if we can't develop a model we can deploy into the clinic to tell patients that once they've undergone therapy, you're going to be okay. You don't need anything more. And so that's exactly what we did. So I met this one young man, Yup DeYoung at the time. He's was a PhD candidate, now an MD urology resident. But anyway, we came up with this project we wanted to work on with this non-coding RNAs, and we basically repeated TCGA and published this in Genome Medicine. So you can see here we developed a 4-cluster solution using an independent cohort. So this is the NAC cohort is a chemotherapy cohort. So these patients all underwent systemic chemotherapy. And we found this interesting 4-cluster solution where one group of patients was doing very, very well. And what was interesting is that our LC3, or this green line here, these patients were predominantly of luminal papillary nature. So they were largely enriched in this luminal papillary subtype. And when we decided to like lump them together, you can see that these patients actually do extraordinarily well comparatively. So at 5 years, 60 months, we have about a 20% death rate or 80% probability of survival, which for muscle-invasive bladder cancer is actually very impressive. So we're very excited about this. We started looking at clinical pathological characteristics, and this just means that we're looking at the clinical data to see if there's any kind of difference between our groups. And you can see that these patients in the LPLC3 group were actually like less likely to have advanced disease. So they were less likely to have PT3 or higher tumors or have nodal involvement. And this nodal involvement and like higher than PT3 is a sign of really bad cancer. So these patients were actually had less aggressive disease clinically. What's also interesting about these patients is they had higher expression of luminal markers. So luminal markers are generally associated with good bladder cancer or less aggressive bladder cancer, and they were depleted in other markers that would be more associated with more aggressive cancer. And you can see again when looking at different pathways that depleted in things like EMT, but enriched in FGFR3 activity. So again, these are signals of good cancer, relatively good cancer, and of course it's true across 2 different cohorts. So this is the result of one of the classifiers we actually built, showing that we can build a single sample model using these data sets. The training testing is NAC and TCGA, and our validation is actually an external cohort of radical cystectomy patients where you can see that the model predicts patients who will do very, very well. So we had, I think it's 10 patients and only 1 event across 5 years. So these patients are doing great. To confirm this, we actually published a study not so long ago, which validated this model in another cohort of organ confined, so clinical T1, T2 patients. And again, we see the same result where these model positive patients or link RNA C3 patients do very, very well. So hopefully that convinces you, that we can actually do this. And I'm going to show you more a little bit of how we go about doing this. And this is moving down the spectrum of disease from muscle invasive back into our non-muscle invasive bladder cancer setting. So it's a little bit different. A lot of this work is done by 2 very brilliant co-op students in my lab, Wendy and Rachel, who have contributed enormously to getting these classifiers put together. And they can answer questions probably better than I can, but they're not here. So anyway, non-muscle invasive bladder cancer, we regroup it clinically into 3 groups, low, intermediate, and high risk. You really don't need to worry too much about the clinical parameters here, just to understand that there are 3 different risk groups clinically. So the clinician coming in will look at a patient and will assign them a risk group based on various clinical parameters. So this is standard of care workflow for clinicians. We want to give them a little bit more information, of course. And so others have attempted to do this using mRNA expression. So this is a group called Uromol. They did this very lovely study here where they have recurrence-free survival curves and progression-free survival curves. And you can see, we're getting some separation, but it's not super impressive. Like clinically, you probably wouldn't do anything differently if you had given this information when you're treating a patient. Other groups maybe a little bit more success, but I mean, dangerously overfit in the case of this study, and this one actually didn't validate in our own hands when I was with the company, but says there's signal there, so that's great. So there's some signal in non-muscle invasive bladder cancer. We can leverage it. Maybe we can build a model, and so that's kind of what we were going to go do. So at this point, I said, let's identify a clinical challenge of relevance, and this comes back down to the earlier slide I was talking about, like what's our clinical challenge, and so. One of the understudied disease settings in muscle and bladder cancer is TA low grade. So this is very early bladder cancer. These are small little papillary tumors. They tend to come back a lot. They don't really progress and they're less likely to kill you, but the coming back is the problem because the patient needs to go experience cystoscopy again and again and again. We don't want that for them because it's very unpleasant. It's also very costly. And so the biomarker challenge then becomes like, how do we develop a classifier to accurately identify these unfavorable or highly recurrent TA low-grade patients? And so, this kind of goes out a little bit out of order, but it's nice to make the story. So the first thing we tried is training to a hard oncologic endpoint. So in this case, we're looking for a binary classifier to predict recurrence. This is a yes, no, 0, 1 sort of question using gene expression data. So we had a training cohort of 276 patients, validation or testing set. We say validation in clinics. Data science types would say testing. They're both technically correct. With 73 patients. And you can see here that, our ROC is pretty abysmal. So Brad here, he's a little like lab icon. He's kind of a little perplexed because, normally this should work. In principle, it should work. It doesn't. So then we tried something else. We're like, okay, now we have a time to event kind of question. We're treating it like a binary problem, but it's not actually a binary problem because, there's a time to recurrence. Some can happen earlier, some can happen later. We weren't accounting for that before. So we're like, okay, fine. We'll start doing some Cox modeling, no big deal. So we get the C index of 0.58. So we're looking at about as useful as a coin toss. And now Brad's getting a little upset, as you can see. But part of the problem with Cox modeling is, yes, it absolutely is better suited, and it's a perfectly good approach to try to do this. But there's a lot of problems we weren't able to account for. And one of it's just weak signal. Okay, so recurrence in TA low grade is driven by a lot of different clinical variables, including the treatment of patients received prior, tumor size, if it has recurrences before. And so the gene signatures might be a little too subtle or a little too variable to actually capture these patients accurately. And then secondly, there's a bit of a cohort mismatch between Uromol and Knowles. So the training data set was very aggressive. We had about a 69% recurrence rate versus 40% in the testing data set. So the lower risk patients may have actually been skewing our population. So, like any good data sciences or bioinformaticians, we come at this problem. We say, okay, fine. Training to hard oncologic endpoints isn't working. We're not getting robust classifiers. We're getting kind of distraught here. So what can we do to fix this? And I mean, we go through the obvious, right? First, I'm going to grab clinical variables into the model and account for the variance. The reason this doesn't work, because we have to think about our endpoint again. Who's going to be using this? A clinician. Clinician is very comfortable with their clinical variables. They don't want you to tell them how to integrate them into the model. They want to keep these independent because they're already making a decision based on them. So we're adding redundancy there. Okay, fine. Increase sample sizes, more data. What more data? We have to go retrospectively collect a whole bunch more patients, generate new expression data. So we can't do that. Imaging and pathology features, fantastic. Definitely a great idea. Again, we don't have these, so we can't really do them. And so finally, we come to the conclusion, maybe we can enrich biological subgroups of patients who have higher recurrence probabilities. And so now Brad's sitting here thinking, okay, maybe we can do that. And so that's precisely what we did. So we took a cohort of 286 patients from Uromol, and we actually we selected the topmost variant link RNAs, and we did consensus clustering. And you can see here that we got quite a clean solution, particularly with link RNA with the link RNA cluster 2, where the typical patient membership ratio was very favorable. A lot of the patients were remaining within cluster 2 and not jumping around with each iteration throughout the bootstrapping. When you look at it more at high levels, these are the link RNAs. So this is just a heat map, and the point of this really is just to show you again that there's structure in the data. So you can see that these link RNAs in the center part below the red bar here are either lower or higher expressed compared to the rest of the cohort. It's not super important what they are. It's just that these very highly variable link RNAs are quite differentially expressed, but consistent within each cluster. Similarly, the coding genes do something very similar. So you can see again this great structure in this data, particularly around the cluster 2. And so we found that these patients, these clusters actually had very distinct gene expression profiles. Particularly cluster 2 had higher signature scores associated with proliferation, such as G2M and E2F. This just means that these tumors are likely growing much quicker. Higher FGFR3 activity signature, meaning they're more FGFR3 dependent, which is very consistent with bladder cancer, but lower immune infiltration, so they have a very distinct biology. Clinically, they have very distinct recurrence-free survival outcomes. So on panel A, you can see that the patients in red, this is our cluster 2, they're doing much worse compared to the other patients. This is just a comparator to another subtyping model just to see how well we're doing, and you can see we're doing a bit better. But it got really interesting when you start to lump the patients together. So if we compress the other three clusters into one group, now we start to see very clear, very meaningful separation for both recurrence-free survival and progression-free survival. And again, remember that these curves, the higher up the curve, the better the patients are doing. The lower the curve, the worse they're doing. When we do MVA, so this is basically taking into account clinical parameters, including age, sex, EAU risk. So this is the clinical risk score and our lincRNA cluster. You can see that for both recurrence and progression, they're both significant on MVA. In fact, progression is very impressive at 4.6 for the hazard ratio, even though the confidence intervals are admittedly wide. It's partially because we just have smaller data sets. And we did remove the high-risk patients here, so these are clinically high risk. And part of the reason we do this is because you're not going to tell a clinician that their high-risk patient is high-risk genomically, because it's just not giving them any new information. But we do leave them in for the clustering in the original sense of it, because it does help guide the clustering solution. So you can pull patients that are like clinically lower risk into a higher risk category genomically if you leave some high-risk patients into your initial clustering. Okay, so now that we have labels, now we're starting to think about how we build an actual lincRNA-based classifier. So the objective here was to build a single sample model based on lincRNA clusters that we have as labels, using, again, the training set of 276 and the validation set of 72 patients. And the primary endpoint for this particular model will be recurrence-free survival. This is the clinical characteristics of the cohort. So you can see that largely they're very similar for age, for sex. Recurrence rates of events are quite different, as I alluded to before. So you can see the rates are quite different. Progression as well. We have no progression in Knowles at all. So Knowles is a much less aggressive cohort clinically. Follow-up time is about the same. And one important caveat is that they are actually created on different platforms. So one's RNA-seq and one's microarray. So this is normally a big no-no for model development, but I'll show you it actually worked, and I think that speaks to the biology and to the difference in these patients' tumors. So the way we went about doing this is we took the uromol and Knowles cohorts, and given their different platforms, we used quantile normalization to kind of bring everything down to the same level. We used MADS to identify the top 3,000 lincRNAs, and then we used nested cross-validation with 5 outer folds and 10 inner folds to get a total of 50 iterations, resulting in, I guess, 179 feature model with about 116 non-zero coefficients. And you can see here that when we ran it on the validation and the testing set, that our cut point, which we used in index to select, was actually quite valid. I mean, most of it either fell far above or fell far below. So we're pretty impressed with this, actually. When we look at this clinically, you can see here that the patients with the model LC2, because M for model, LC2 patients actually did much worse. So we had 7 patients we identified. Out of those 7 patients, we had 6 events compared to the rest. This is actually quite striking, considering this cohort is fairly non-aggressive. On MVA, we can only account for age and sex, unfortunately, in this MVA. We found that the hazard ratio is an impressive 4.6 with a significant p-value again, but very wide confidence intervals. And finally, because we didn't have any sort of ground truth, we couldn't use our standard ROC or AUC approach for testing sensitivity specificity. These are biological classes, so it makes it a little bit more challenging to actually get those kind of numbers. We instead opted to use biological validation. In addition to the clinical validation, you can see here that these patients in both Knowles and uromol. So uromol is the training set, Knowles is the testing set. That the pattern of gene expression for the signatures is very consistent across both, resulting in patients that have higher proliferation with LC2 for both Knowles and uromol, but also they have higher FGFR3 expression profiles as well, and depleted immune signature profiles. So the clinical implications here, I think, are maybe many, but really what we've developed is a clinically robust single sample classifier identifies these high-risk patients. So we're actually able to take patients that are clinically lower or intermediate risk and put them into a higher risk category where they may get more aggressive therapy or more frequent surveillance. So they might have more cystoscopies. And the plus side, of course, if you're not model positive LC2, you might get less cystoscopy because you could be more confident the patient isn't going to be advanced. And unfortunately, this is a relatively small patient population, and we are training testing on two different cohorts developed on two different platforms, but I think that the biology spoke pretty loudly here. And, you know, you can see Brad here is giving a little flex because he's quite pleased with himself. So should Wendy, because she actually built this model. So bring it all home, really. Non-coding RNAs, like link RNAs especially, are very pervasively expressed across many biological systems. And I hope I've kind of shown you that, you know, they are expressed differently across cells and tissues, and we can leverage these as biomarkers. They're a powerful tool to use for biomarker development. And Dutch elm disease, you know, the fungus, it's very easy to do something very simple with PCR. You could say yes, no, big band, small band. Super easy for fungal strain typing. Bladder cancer, a little bit more complex, obviously. We need more complex tools. So in muscle invasive, I showed you some data around link RNA-based classifiers that have robust performance in identifying luminal favorable tumors. So these are the good patients. And non-muscle, we have used it to do exactly the opposite, find our aggressive patients with immediate clinical implications and helping select patients for more aggressive therapy or increased surveillance. And if nothing else, hopefully the take home that you got from this is that we can actually use long non-coding RNAs and take this so-called genomic noise or junk DNA and turn it into clinically useful biomarkers to help stratify patients for risk. Of course, you know, there's always an army, and I mean, there's so many people to thank for various phases of this work. But you know, in my own group, so Wendy and Rachel have done like lion's share of the heavy lifting and developing some of these models. Robert Bell is in our audience right now as a biostatistician with Colin Collins' group, has been super helpful in getting these things off the ground as well and entertaining my students when I'm not in the lab with them. Peter Black, of course, our champion at the VPC and department head of urologic sciences. You know, he basically hired me, so I have to thank him for that. And collaborators like Joep de Jong, a very close friend and close collaborator, Netherlands, Lars Juskot, he's helped me get hold of the Euromol data set, and we do a lot of work together in various capacities. A number of Veracyte people who helped build the original link RNA classifier for the muscle invasive disease. And then a whole smattering of collaborators and urologists I've worked with throughout my career have really helped build a lot of what you've seen today. And before I leave you, I'll just do the shameless plug. So I'm only been here for about a year, so I am recruiting. I think if you're interested in the intersection of data science and clinics, this is probably a great opportunity for you. You will interact with clinicians, you will interact with data scientists, and build things that kind of cross both worlds. So we're looking for basically anybody, grad students, postdocs, anybody with an interest in this type of work. So if you think you might be interested, you know, send me an email. That's my lab website at the bottom there. It's in development, so it's not super pretty, but we're getting there. It's a little grim right now, but that's okay. Anyway, that's me, and thank you for your time and for coming, and I'm happy to take your questions.
The microphone, so it's just
Yeah, for Dr. Cox always help you.
it's just red. If it's flashing, you have to wait.
So I guess.
Oh, wait, so you can go ahead. It's not flashing.
Oh, okay. So first question, so I have a few.
Sure.
So you're using the long non-coding RNA as the biomarkers, right? So
Correct.
do you have time to like investigate what makes those long non-coding RNA different from the other three clusters? The one cluster two.
Oh, you want to know like which specific link RNAs? Well, I can show you. I mean, I'll show you something. I'll give you this thing. So, I mean, there's not a lot that are known for various reasons. Like, but UCA1 is like urothelial cancer associated long non-coding RNA, generally associated with better bladder cancer. So it's actually not very highly expressed in our cluster 2. We haven't really done a lot of like detailed analysis into which ones are different and why they're different and whether that's interesting or not. That's a little outside of the scope of the current research program, but definitely on the agenda for downstream once we get our feet underneath us.
So now that you have a pipeline, have you considered applying it to different cancers and see if
That was the basis of my Canadian Research Society prostate cancer grant, actually. Yeah. So we're applying a similar approach to what they call a grade group 2 prostate cancer. So this is like an early stage disease where treatment decisions are very challenging because they don't really know how aggressive it is. So some of these men will end up on active surveillance. Some of them will get definitive therapy, meaning like hormone deprivation or surgery. And so we're trying to develop a similar classifier for these patients that can help distinguish them. Preliminary data suggests that this works very well. We have I think it's 3 or 4 cluster solution that ended up having very distinct biology and some very hand wavy association with clinical variables because we didn't have what we were looking for in our actual discovery cohort, but we applied it to a different cohort with only had 11 patients. So the associations are like n equals 1, n equals 2. But there's some indication that there's some differential clinical variables for those two. So yes, definitely. Yeah.
And lastly, and you mentioned this earlier, like the turnaround time for this and it's quite important, right? So
Yep.
I'm guessing it's more or less just the sequencing and the clustering is pretty quick. It's just
So the classifiers are actually once you have a classifier, it can be very quick. It's just a matter of getting the tissue sample to the lab, getting it processed, RNA sequencing done, getting the sequencing back onto the computer, getting the algorithm to run and spitting out a result and getting it back to the clinician. So part of the plan, the long-term vision for the lab is to take these biology or link RNA-based classifiers. If we can model them, we get a classifier that works robustly. Can we then translate that to a digital pathology model? So it's kind of like using that as a label to move the science forward, move the test forward, because digital pathology is routinely collected in every clinical lab ever. So you just scan your slide, add your algorithm, done. Doctor gets a result in a day or two. Versus this would take up to 2 weeks.
2 weeks is still within.
Depends on what you're. Yeah, it is definitely within the window for sure. Like I mean, most chemotherapy or BCG decision making will take place within 4 weeks.
No, you're flashing. That means you have to wait.
You have to wait.
Oh, it's at the bottom, so you'll be going this your turn. You have a question.
So I think in like the next slide, maybe the one after that, you look at the association between interferon gamma and the clustering.
Oh yeah, sure.
I just got a particular interest in interferon gamma. I was curious whether if that was related to if there was differences that you just kind of moved through.
Is it? Do you want just the class? This one?
This.
Yeah.
So this is clustering based on the groups, right?
Right.
And so is this showing that the red group is the one that had the worst progression?
The worst progression, yeah.
And is this showing that it's got lower interferon gamma?
Right. Yeah.
Okay.
Yeah. And lower immune infiltration too, because immune 190 is one of those lovely immune-based signatures that just seems to do everything very well. We've correlated with MKI67 expression. It's beautiful. It's like one of those marvel signatures that we got hold of at one point and never let go. But they're definitely less immune infiltrated.
That was awesome.
Yeah, sure.
I was wondering, how do you prevent urinary or bladder cancer and prostate cancer? How do you prevent it?
Well, prostate cancer, if you live long enough, you're going to get prostate cancer. We all will. I mean, you can't really prevent it. It's an inevitability. Bladder is a little bit easier. Smoking, don't do that. Don't be exposed to hazardous chemicals. Try not to be a male. It's like, yeah.
That's my second question. Why is it more prevalent in males?
That's a fantastic question, actually. We published a study 4 or 5 years ago that shows that bladder cancer may have something to do with androgen activity. Just there's an association, it's a statistical association, but I'm not going to say that's a biological association. The good news is males tend to get less Yeah, and then get better bladder cancer, a little less aggressive clinically. Women, unfortunately, or females tend to get more aggressive bladder cancer. Why that is, we don't know. No one had a clue. Like,
See when we take smoking out, because men smoke typically.
they do, yeah, yeah, yeah.
Because of all the presentation, first of all,
Thank you.
But I have a couple of questions. So instead of using different data sets as different sets for training and validation, why didn't you just pull them and then partition them?
Yeah, that's a great question. I think part of the reason we don't do that is because it invalidates the validation of the cohort, because if we split them, if we have them unique, it's because what we're looking to do basically is we want to build a classifier that's deployable in a clinical setting, right, where there's a lot of variability. If we try to mitigate that variability too early, then it kills the robustness of our classifier. So that's largely why we try to keep things separate. Instinctively, that's a good question. Yeah, absolutely. I mean, I think I threw this at Dr. Hatch's class one time in bioinformatics. I can't problem-based learning, right? Yeah, and
Here, so exposed to this question.
yeah, very exposed to this question. I think most people actually did exactly that thing, and I'm like, no, this is not going to work. It's intuitive, yes, absolutely. You want to do that, but it doesn't work practically.
But you want your classifier to be aware of systemic biases.
True.
Right? So by mixing them, you would also expose the system to those biases, and you force the system to not care about those, right?
That's true.
So maybe, like, in briefly, it might not work.
It might not.
Intuitively does.
Yeah.
What I realized there.
It's odd what intuition does to you, right? I mean, intuitively, I mean, I think, and not to throw under the bus here, but I mean, when we were doing this initial classifier development, Wendy said to me, so I can build a better classifier by training to hard oncologic endpoints. And I said, oh, really? Okay, let's go. And that's a lot of the data she showed right there. But I mean, this comes from like a decade of experience working with teams of data scientists who have tried exactly that. Eventually, we had to do this kind of clustering-based methodology to get more accurate classifiers. So yeah, intuition is an interesting thing, though. I mean, it took a long time to develop this approach, this pipeline. So it seems very routine now, but it was actually quite difficult to get where we are. But it's a great question, though. Yeah.
So, let me ask an uneducated question. So you showed the heat map for both non-coding as well as coding RNA, right? And you have to have some separation.
Oops. Sorry, I can make you guys busy. Yes, yes.
So we call it junk.
Yeah.
It's not in fact junk.
No.
Sometimes regulators of the sorts.
Yeah.
And sometimes they may have a downstream effect.
Sure.
Right, on the coding genes. So have you ever looked at potentially an association between the non-coding and the coding part to see that if you can mine potentially, is it just affecting some specific pathway through the non-coding RNA?
Right. That's a very good question. I think, I mean, in my postdoc days, I was very interested in trying to identify long non-coding RNAs that had some biological function, so I could take it to the lab. Computationally, that's actually more hard. It's more challenging than it looks, just because a mere association, statistical association, doesn't imply biological function. That said, I think with this particular study, and given that some of these non-coding RNAs are showing up in the prostate space as well, I think we have good indicator that we might be able to reduce the dimension of these data down to like a handful that could be quantified in the lab for proliferation, at least. I think like a lot of these seem to be proliferation associated, but it could be cause and consequence, cells growing faster, more of these, just noise. But it's a good question. Definitely, something I want to spin out in the lab at some point, maybe in a couple of years once we kind of have some funding and some more people and can do this some fun threads to pull on as an academic. But it's a really good question.
Okay. I had a question what do you think of other sequencing modalities? Because the focus so far has been on RNA, whether they would be fruitful in this case, use or not, whole genome sequencing or genomic markers or or even methylation?
Sure. Yeah, that's a good question. I think I'm a bit biased. I've lived in RNA basically my entire career. What I like about RNA is that it reflects the state of something, granted in a snapshot of time, whereas mutations sort of say something happened, right? So mutations like, oh, there's a mutation. Great. We don't even know if it's expressed. We don't even know if it even matters. It could be mutated as X ways from Sunday. It doesn't really matter. Never does anything for the cell. What I like about RNA is because it's so reactive, it's so like dynamic, is that it does give you something to get a hold of. And I think that's why I prefer RNA. Methylation kind of falls in the same similar bucket, right? Maybe methylation would be a little bit better because it could indicate which promoters are available, but I think RNA is kind of the sweet spot. Although, I get it, it's finicky. It's a pain to get it, we're working with formalin fixed tissues, so we're working with very degraded RNA most of the time. It's not favorable, but you get signal like this, you can't really argue it's working.
So I guess I'm wondering, if you can just comment a little bit more on what you think the mechanism is, like whether the long coding RNAs are actually doing something. If they're like maybe like a surrogate marker for something else, like they share promoters with other RNAs and like so if it's just like they're being promoted because something else is happening, or whether they're doing something themselves.
I like the idea of them being a surrogate for something else being expressed. Like they share a similar promoter, say with MYC or something like this. And so they're just amplifying signal and it allows us to capture that signal. I'm not super convinced that all of them are doing very much of anything. Otherwise, a lot of these GWAS studies and things would have revealed something. I tried doing some work with a non-coding RNA one time and, doing pull downs and reverse pull downs and like seeing what bound to it. And it bound to RNA binding proteins. It's like, well, yeah, shock me, right? Like, so I don't know. I don't know if they're actually doing all that much, but I do think, the cell is very noisy. We all think about the cell being super regulated and so structured and tight, but a lot of it is just patchwork to fix problems that it created earlier. It's like, oops, I overexpressed this. How do I downregulate that by overexpressing something else? And so I think that creates a lot of noise, especially in a cancer setting. And I think that the noise is just getting amplified, and that's what we're seeing. Some questions.
Great presentation.
Thank you.
I think in one of your last slides, we mentioned that you didn't have ground truth. So did you add ground truth or just use those clustering labels as you would?
Yeah, that's all we can use, but then we can't really run sensitivity specificity on our training cohort because we use that for training, right? So even though that would be truth, it'd just be overfitting. Yeah.
So your labels were the clusters.
Correct.
So did you use the same data set and features for clustering and training?
I'll make sure I'm gonna get this right because I don't want to say overfit it. Like, no, yes, with the same features. Yeah, I think if I'm understanding your question correctly.
So doesn't that cause like more overfit to those clusters because you have been used those same features to cluster?
But when we do the testing, it was a totally different independent data set, right? Yeah. So that's the problem with the ground truth, right? Once we run out a new data set, we're like, I guess that's correct, but there's nothing we can use to say, yes, this is correct. And it's even worse because it's biology. So there's some noise in there. It's like,
Do we still have time? My question is between the advantages of the long non-coding RNA and the short expressed RNAs. Can you go to the slide that had the comparison of different kinds of these signals?
I mean, sorry, guys. So
Doesn't matter. Do you think is the long non-coding RNA better to classify compared to shorter or like protein coding RNA, or is it just that those survive the tissue fixation better? So it's a signal that you can still pull out.
I think it's like, because we try with coding genes often, like a lot, the actual classifier we built was a composite of coding and non-coding RNAs. The labels are generated exclusively with non-coding, but the actual classifier was built on both. For generating, like, I guess what you could call labels. I think that the specificity that they have to whatever disease state, in this case, like aggressive cancer, is so high that they actually were able to delineate these aggressive patients better than mRNA alone. I mean, we have tried composites of using both, but for whatever reason, the non-coding RNAs just come out loudest. They're just so yes or no. I couldn't tell you why that is, though. It's very curious. It's
Are they expressed in healthy tissue?
yeah, absolutely. Yeah, yeah. Yeah, they're not universally disease specific. They're just like they're amplified in disease or I mean, so I mean, well, X chromosome, for example, exists. This is one of your classic non-coding RNAs. It literally is essential for female development because it silences the extra X chromosome. It just, coats it. So it's kind of cool. So they do stuff, but how many of them are doing so?
Compared to coding RNA, does the long non-coding just get like transcribed more?
No, I wouldn't say that. No, but probably about the same. I mean, if you look at this graph here, the ratio is not abnormally different. I mean, they're there. It's very strange. Like it's like there's not. I don't have a definitive answer for you. I don't actually know.
Actually, I was from, I have a lot of bioinformatics experience.
Okay.
But I am more familiar with like gene expression models, right?
Yep.
And a lot of the validation that you use in RNA-seq shows that there is a significance for cluster 2.
Yep.
So I guess I'm wondering why not use those sets of genes and build a model off of that and use the RNA-seq as a way to diagnose instead if it's more commonly used.
So you're thinking maybe just grab like a subset of genes and just use that, like say a 6-panel marker.
6 might be a little low, but
Yeah. Well, I mean, okay, so these box plots here, they're all like get to them. Sorry, you guys, I know you're gonna get a little dizzy. Don't want that one. I want this one. This is what, so you can actually see, I can't zoom in, but you can see that the distribution of the actual individual data points in each group is not, I mean, they're not that wide, but they're fairly wide, right? So there's like each patient may be doing something slightly differently. So some may be super proliferative and then maybe lower in the FGFR3, and some may be like more FGFR3 and less proliferative, but that balance across all our different genes within the actual model itself, the 172 features, whatever we put into there, is like important in grabbing those patients. This is just a biological readout of a classification, like if that makes sense. So I don't think you can really take that then and put it back and use that as the model. It would collapse on itself. But it's a good question. I mean, who else wants to ask a question?
So I'm just going to ask one quick question and I won't come in. So what does data that you use in general, right? So you use RNA-seq. And these are like gene expression data, like this is mRNA. What are you using? Is it total RNA?
Yeah, this is total, for URML is total RNA, for DOLS is total RNA. So this is all fresh frozen tissue.
So I was talking about economics of this, right? So you need to do a lot more reads to generate these samples.
Right.
So would it be clinically viable? So in gene expression now, you just
Sure.
put on $1, you're just getting data done. So what we're I think it's going to be 80 million rates, probably much more we need to generate as a number of reads.
Right.
So is what goes back to like a panel. So as soon as you have a classifier, do you think that we should just do a panel actions if we become on this?
Yeah, that's an interesting question because, Decipher, so GenomeDX, the company I joined, originally was considering that, and several of the competitors like Oncotype will do this. They'll have a panel of genes. The strength in using sort of the genome-wide or whole transcriptome approach, in Decipher's case, these microarrays, which actually work really, really well in FFP, very tolerant to sequence mutations or damaged RNA. The reason they used that was because they wanted it downstream for further classifier development. So they created essentially what was a resource. In fact, we're actually with Nada's group in a discussion with Decipher about looking at one of our signatures we're developing for ARV7 prostate cancer across 100,000 transcriptomes that they've accumulated over the last decade or so. So that's why they like whole transcriptome, why we don't do panels, even though, you know, technically cheaper, faster, easier. You just lose so much.
And we need for further down, but biology, and
Yeah.
we need to cost. So finally, you advertise for the lab. Are you going to give out Brad with new students coming to your lab?
I'm gonna give you stickers with a Brad on it, and you get to go to cool conferences. Like if you want to do, I mean, there's one bladder cancer conference that I think everyone should go to because it's just really, really great meeting. But like, I mean, for machine learning types, obviously they could go there.
I think with this one, we can close up question. Do we have any questions from the Zoom? Okay, so let's thank Dr. Gibb one more time.